Okay, so here are our notes on mutations. So if you haven't already, go ahead, open up your assignment here called Mutation Notes. Your copy of your Cornell notes should be on here. And make sure that you're doing some highlights and a 3 to 5 sentence summary before turning in, and then create a link of your completed notes on page 47 of your notebook. All right, so let's go ahead and get started. Okay, so we're going to talk about human genetic mutations. So what are the different things that can cause a mutation in our genes? And remember, those mutations are like a change in a word of a sentence. And so it causes the proteins that are made when we talked about protein synthesis to not make sense, right? It makes it a totally different protein, okay? So our essential question for this is, what are mutations and what do they do to the DNA? So make sure you put that at the top of your notes, okay? All right, so there's two types of mutations I'm going to talk about here in these notes. The first one is chromosomal mutations, which means it's in the entire chromosome, so a long strand of DNA. And then specific gene mutations. So gene mutations means within a specific gene. So like our letters that we talked about, like uppercase B or lowercase b, there's more than just one little set of codons in there that codes for one gene, okay? So we're going to talk about a whole chromosome, and then we'll talk about some just specific gene mutations can happen. All right, so what are chromosomes? Let's remind ourselves what chromosomes are. So this right here is a normal karyotype. This is what we call a karyotype. So we can actually do this. This is someone's DNA. These are all their chromosomes, and we can actually have them line up. So you can see these are our sister chromatids here, and we can type them, okay? This is the X chromosome that codes for a girl. If there's two X's, it's a girl. And if this is the Y's are a little bit smaller, okay? And we'll see what those look. But you can see there's two X's here. So I can tell, since there's two X chromosomes here, then that is one of a girl. Okay, so this is a normal human karyotype chromosomes. A karyotype is just a picture of chromosomes lined up to look at and compare. So we can look at how long they are. We can see how they match. You see they're kind of different colors. They kind of get really pretty. Okay. Humans, remember, have 23 pairs of chromosomes. Remember when we talked about that in mitosis? With one chromosome from each parent. So remember, our gametes, when we talked about meiosis, were haploid, and that's why we only have 23 pairs, because we only have half of these, right? We have a total of 46 chromosomes, so 23 times 2 pairs is 46 chromosomes, and that's what you have inside your body. And then when we have our gametes, we get 23, okay? The chromosomes are coiled up in DNA, so in our little double helix. And under normal conditions, all the chromosomes are inherited, are intact. That's in normal. And we're going to talk about what happens when there's problems, okay? So a chromosomal mutation is any change in the structure or number of chromosomes. So I just said each gamete should have 23 chromosomes, single chromosomes. What happens if we have 24? What happens if we have 22? Okay? And on a large scale, it affects many genes. So there's thousands and thousands of genes on each of these chromosomes. So now we're affecting more than just one gene. Okay? And there's 5 types we're going to talk about. The first one is deletion. The second one is duplication. The third one is inversion, then translocation, and non-disjunction. All right, let's talk about the first one. This is chromosomal deletion, and it's just as what it's named. It's deleting one of our genes. So we can say we have here A, B, C, D, A through G. These are all our genes. And then what happens is C and D get deleted. We don't know why, it just happens. So this was what we would call a chromosomal deletion, which means our chromosomes are shorter than they should be. So when we do those karyotypes, we can see that it's shorter. And what are the causes? What do we see come out of it? One of them is called Hishorn syndrome, which is a severe mental retardation, and Karyotype syndrome, which is mooing sounds and mental retardation. So it causes a lot of problems in the brain, but it also causes problems with voice, with processing, with the way they look. There's so many different genes that are actually affected when you actually delete some of them, okay? The second one we can see is chromosomal duplication. So it's just like it says, you're going to duplicate the same genes. So again, we have a normal chromosome, A through G, and now we have a longer one because now what happened is C and D got duplicated. So instead of having A, B, C, D, E, we have A, B, C, D, C, D, E, F, G. So there's extra chromosomes in there. And so this is what happens in duplication. And the causes, or what we see come out of that, is called Charcot-Marie-Tooth disease, which is you would have a high arched foot, claw feet, and confined to a wheelchair. So that affects really their mobility, the way their legs develop, their feet. It causes so many different issues with how your body grows. Okay. The third one is called chromosomal inversion. And if you think about it, remember when you do math, when you do the inverse of a fraction, you flip it, right? Same thing here. So what's going to happen, instead of going through our nice little alphabet all the way to G, what happens, as you can see, is that we get a switch. So instead of going A, B, C, C and E get flipped, right? So now we have A, B, E, D, C, F, G. So the inversion is that it gets flipped upside down. So this little thing, instead of C being here, D will stay in the middle. E will flip over, and then we'll have D and then C. So it's just an inversion of some of the genes. And so what can happen there, as you can see here, if you have blue and red, then what happens is this portion of the red gets switched to the blue, and this portion of the blue gets switched to the red here, okay? And one of the things we see from this kind of chromosomal mutation is called four-ring syndrome. You have a cleft palate, club feet, and then testes don't descend. So you can see all the different organ systems and features of a person that can be affected by just flipping them over, inversing the genes. Fourth one is called chromosomal translocation. So think about translocate. Location, we're going to take the location of some genes and we're going to move it all the way somewhere else. So genes are swapped with non-homologous chromosomes. So it's like you're translocating or moving something to a completely different chromosome. So we're going to take the genes, okay, off of here, so there's blue, and we're going to flip it all the way onto a non-homologous. Remember, homologous means the same. So a chromosome that is not the same. So the genes get sent to a totally different chromosome. So you can see here on chromosome 3 here, there's a piece that got translocated, and the little piece here ended up all the way down here onto this chromosome. So they flip-flopped. And some of the causes, or what we see from this, when this happens, is called Burkitt's lymphoma. So this is a cancer of the lymph nodes, and it happens most of the time in children from this kind of mutation. All right, this is probably one of the most common chromosomal mutations that you see out in the world. Chromosomes fail to separate. So remember, during meiosis, we had those copies of the chromosomes here. We had 2 of them, and they need to separate for that law of independent assortment, right? So we only have 23 in each gamete. What happens when they don't separate and they end up all here? So this cell is here missing some, and this has extra. So this actually happens in meiosis, okay? And it can happen in meiosis 1 or meiosis 2. So remember, it goes through the same steps in meiosis 1 and meiosis 2. And so non-destruction, or where the sister chromatids do not separate, can happen in either one and cause the actual gamete to have more than 23 chromosomes. Okay. And so non-destruction, meiosis produces gametes, and therefore a baby with one missing. So you'll have what we call monosomy. So they have one missing chromosome, right? Mono means one. Or you'll have a baby with extra chromosomes, and we call that trisomy. So you can see there's 3 copies of a chromosome instead of just the one, okay? And we see this most of the time when we see it here in meiosis 2. So you see yellow and blue, okay? Yellow and blue get split up just like they're supposed to. And then the two yellows get split up nicely, but the blue, instead of splitting up nicely, it ends up all in one cell, and this cell has none. And so you have problems with both of these cells, okay? And so then when something gets fertilized and it has two copies of it, then it comes in and now we have three, okay? And this is what we call a trisomic zygote. So a zygote is another word for baby. And this is what we get when we don't get that separation, okay? When we get that non-disjunction. And so every cell in that baby's body will have three copies of whatever chromosome didn't separate instead of two. And this condition is called trisomy, okay? So now they have three copies. Crazy, right? Because you get one from the dad, and two from the mom, or two from the dad and one from the mom. So that you get three copies. And one of the most common ones that you will see around is called trisomy 21. And that just means individual has three copies of chromosome number 21, okay? And this is what you would see as Down syndrome, okay? And so children who have Down syndrome actually do have the three copies of chromosome 21. Klinefelter syndrome affects only males. They have an extra copy of the X chromosome. So usually males are XY, and so the Klinefelter syndrome is an XXY. So they have an extra copy of that X chromosome. It does not split apart, okay? And so you also have different versions of this trisomy. So there's trisomy 21, which is what we mostly see. There is also another one that is usually they don't survive. It's called trisomy 16. And my sister-in-law actually had a baby that had trisomy 16, and that baby did not survive more than an hour after it was born. The organs were growing outside the body, and so that baby died. And that's what happens with a lot of these chromosomal mutations, okay? And like it said, most chromosomal mutations are lethal, which means those children do not survive, okay? And if the fetus survives, they tend to cause widespread abnormalities. So they have a lot of problems throughout their entire body. And the example that we can think is Down syndrome, which is that trisomy 21, okay? And this is the cause. They're super cute, though. They're non-disjunction of chromosome 21. And they have three copies of chromosome 21. And that's what causes some of the issues and the features that you see on a Down syndrome. And I did a lot of research on Down syndrome for a family. And there's just a lot, even mentally, as far as focus, as far as memory, all of those things are affected. Okay. And remember, we talked about kerotyping. What is kerotyping? I just kind of want to remind you. A kerotype is a test to determine the number and appearance of chromosomes. So they can do a kerotyping of a baby, and they can already see that has trisomy 21. Because at chromosome 21, instead of having those nice two copies, there's three. Okay? And they use that kerotyping to determine chromosomal mutations in an individual, and can be used to test for Down syndrome or Klinefelter syndrome, and that's what they do in the amniocentesis. Usually they measure the baby for a long time, and they think, oh no, this one we might have some issues. It's not growing as normal as we would like to see, and they'll do these tests, and they'll do the kerotyping, and they'll find some of these mutations, okay? This is an example of a kerotyping for Down syndrome. So you can see here's all of our chromosomes, nice, okay, 22, and then this is a girl because we have X and X, and so that's our 23, but you can see at 21 we have three copies, okay, and so that's when we can know, okay, this child has Down syndrome, okay. And we can do the amniocentesis that I just talked about, and what they can do is because the DNA of the baby is in the fluid around the baby, we can stick a needle in there, get a little bit, and then we can go ahead and test them and run the DNA to get that kerotyping. Okay. So key point number 1, too much or too little DNA is bad. You always want the exact amount that you always need. 23 chromosomes. That's it, and they should be in the right order and not be mixed up, okay? All right, so let's review. What are the 4 types of chromosomal mutations? I hope you guys said, right? You said inversion, deletion, right? You've got non-disjunction. Got non-disjunction. You've got translocation, inversion, duplication, deletion, right? So these are the 4 main types, and then non-disjunction is our 5th. Okay? All right. And it says, which type of mutation is probably most damaging? Really, they're all pretty damaging, but most of the time that trisomy is the one where we see the highest death rate. But everything else, pretty much, a lot of times you see a high death rate, and all of them can be equally as damaging. All right, so let's go on to gene mutations. That means instead of the entire chromosome, instead of the big long string, we're going to go to just one. So remember when we talked about our base pairs? So now we're going to talk about just one single one. So it's just a small scale. It's not affecting many genes, just one. And it's any change to the DNA sequence of a gene. So nucleotide bases may be added, missing, or changed, just like we talked about with chromosomes, okay? But there's 2 main ones. The first one we'll talk about is point mutation. What does that mean? It means at one point, one base is changed. So instead of having an A here, it gets changed to C. And remember, now we're going to change that codon, and we're going to completely change the amino acid, which completely changes the protein. Remember the sense. If we change even one letter in a word, it changes that word, which then changes the sentence, okay? And then we have what we call a frame shift mutation. And we can have 2 of them. We can have an insertion where we have the T come in from nowhere, an extra one in there. Again, changes our codon, changes our amino acid, which changes our protein. Or we could have something deleted. And then again, that changes our codon, which changes our amino acid, which changes our protein. Okay? And so point mutation is one base, is substituted for another. One of the causes that we see, or one of the things we see from this is called sickle cell anemia. And sickle cell anemia just basically is, instead of our blood cells being a nice big round shape that oxygen can really bind to, they're kind of concaved in and half kind of crescent moon, and they can't carry oxygen as well, okay? And then there's 3 possible consequences. So nonsense mutations, it could code for a stop, which can translate the protein. What we call a missense mutation, that codes for a different amino acid. So just changing this one base, you could end up coding for a stop and stop that protein where it shouldn't be, maybe in the middle of the protein. You could code for a completely different amino acid, and so you're completely changing a total word in the sentence. The first one, I guess if I use the sense, is like you are putting a period in the middle of a sentence, okay? And then silent mutations is it still codes for the same amino acid, and you don't really see too many effects from it because the same amino acid has gone in, okay? All right, a frame shift is when you're doing one or more bases, so not just one. So a point mutation is one base. A frame shift is more than one, okay? Are added or deleted. And one of the things we see, one of the diseases we see from it is called cystic fibrosis. And that's where they really produce too much mucus in their lungs and they can't really breathe. And it can be caused by an insertion adding a base or deletion removing a base. And so doing that can cause the amino acid to change, or like we just saw in, it could also code for a stop and stop that protein being made in the middle of the sentence, okay? So what are some causes of mutations? Why do they happen, right? Well, the first thing is spontaneous. It just happens. We have no idea why. It can occur during DNA replication. Remember we talked about the cell cycle, and we talked about there's different places where there's checkpoints. And so in DNA replication, that checkpoint could have been missed, and then that cell goes on. And it could also be caused by what we call mutagens. So the physical mutagen could be like radiation from UV rays, X-rays, or extreme heat. So that's why we wear the lead when you get an X-ray, because it can actually cause a mutation in your DNA when it's exposed, okay? And chemical molecules that misplace base pairs or disrupt the helical shape of DNA. So if you have a chemical exposure, kind of like nuclear exposure, that causes all kinds of mutations in your DNA. And one of the things when I worked with mice and I was doing Down syndrome research, we actually made a Down syndrome mouse by actually exposing it to UV and X-rays. And so we took not the mouse, but we took the cells that we were going to use to make the mice, and we actually caused the mutation to happen, and we tested for it. And the ones that did cause the mutation, those are the ones we kept and made our line of mice from. Okay. Gene mutation. So key idea, a mutated gene will make a mutated protein, which is not good. We want the right proteins, okay? And mutant proteins are trouble. They do not go where they're supposed to go, and they do not do what they're supposed to do. And that's why we see all those different problems and issues with these diseases, okay? Okay? And as I just talked about, sickle cell anemia is definitely an example. And so usually our red cells are shaped like a disc, and so we can bind oxygen to it. And a sickle cell, they're abnormal crescent kind of shape, okay, like smashed in one side. And they can't carry oxygen as well, and so they form clumps and get stuck in our blood vessels. And that's not a good idea because we need our blood to flow to take carbon dioxide out and bring oxygen in, okay? So this is an example. So this is just a picture example. So this is what a sickle cell, what a normal cell looks like. You see it's nice and round. It's got a bunch of oxygen in there. This is a sickle cell. It's just flat, and it has less surface area for that oxygen to go. Okay. All right, another example we talked about is cystic fibrosis. This is what we call autosomal, which it just passed on, and it's recessive disorder. So again, it's that lowercase letters, right? And the symptoms are thick mucus in the lungs and digestive tract, and they have constant lung infections and impaired digestion. Okay, and they can test and do the Kerotyping and know if a child's going to have cystic fibrosis as well. And usually the only cure for cystic fibrosis is a lung transplant. Okay. Okay, who gets it? Anyone, and most common it is in Caucasians. Okay, and it actually affects 1 in 3,000 Americans, and 1 in 23 white Americans actually carry the mutant allele and can be passed on. Okay. And so what happens is CFTR gene is mutated, so three base pairs, so that would be a frame shift, are deleted and causes a mutant protein. So you can see, okay, that instead of having a nice, beautiful, long protein, this one's all twisted and convoluted, and so it can't really function as what it should be, okay? So this is what usually is when it happens, okay? So you get the normal ions coming in and coming out easily. That's not what happens with cystic fibrosis. It gets stuck, okay? So our key point number 2 is mutation of a gene equals mutant protein, which is bad, and dysfunctional proteins cause the symptoms of the disorder. We are talking about mutants. We're not talking about X-Men. Not good. We don't get powers. We actually have problems, okay? All right, so now this is the end of our notes. Go ahead and highlight any important notes. Make connections to any of your notes in the left-hand column. Write any questions you may have in the left-hand column, and then use your highlights, connections, and questions for your summary, and then make sure you're writing a 3 to 5 sentence summary. And if you have any questions, please let me know.